Welcome to AP Biology. This video is on DNA technology. DNA technology is the manipulation of organisms or their components to make useful products. Central to DNA technology is recombinant DNA. Recombinant DNA contains genes from two different sources. These sources are often two different species. The DNA is combined in vitro. In vitro means it's combined inside of a test tube. Recombinant DNA is central to genetic engineering. Genetic engineering is the direct manipulation of genes for practical purposes. If you look at these two pictures here, these two different species have been genetically engineered. Here you see genetically engineered mice, and here you see genetically engineered fish. Both of these species contain genes which originated from another species. Here you see these genes allow these organisms to bioluminesce, to glow in the dark. So both of these species contain recombinant DNA, DNA that contains genes from other sources. How do you create recombinant DNA? Recombinant DNA is created using restriction enzymes. Restriction enzymes cut DNA at very specific sites. Restriction enzymes are also called endonucleases. They're also called nucleases. Restriction enzymes are obtained through bacteria, and they're often named based upon the bacteria that they've been found in. And these restriction enzymes are going to cut the DNA at very specific sites that we call restriction sites. These restriction sites are often palindromes. If you look at the two strands of DNA, they are the same sequence forward and backward. When the restriction enzyme cuts the DNA, it can create two types of ends. It can create what's called a blunt end, which means that it cuts it straight through, or it could create sticky ends. Sticky ends are what we see right here, and they are staggered. In genetic engineering, we want to create sticky ends because the sticky ends can be recombined with other DNA. Once a piece of DNA has been cut using a restriction enzyme, that gene, here you see our gene of interest right here, can be inserted into a new organism's DNA. In order to insert this selected gene that you see right here, we cut the organism's DNA with a restriction enzyme, and that gene is inserted using the enzyme DNA ligase. Here you can see our end result. This is a recombinant DNA molecule. It's a DNA molecule that contains a foreign piece of DNA. In this case here, the gray gene is the foreign piece of DNA. It could have even been obtained from another species. In order to insert genes into organisms, we need to use what we call vectors. The vector is the method of transferring the gene from one organism to another organism. In genetic engineering, we often use plasmids as the vector. We use plasmids because they can easily be inserted into bacteria through the process of transformation. Here we see a short animation showing us how restriction enzymes work.
Restriction enzymes are enzymes isolated from bacteria that recognize specific sequences in DNA and then cut the DNA to produce fragments called restriction fragments. Different restriction enzymes recognize and cut different sequences of DNA. This restriction enzyme recognizes the DNA sequence GAATTC. It cuts the DNA strands between the bases A and G. The resulting single-stranded sticky ends have the base sequence AATT. This restriction enzyme recognizes the DNA sequence CTGCAG. It cuts the DNA strands between the bases A and G. The resulting single-stranded sticky ends have the base sequence ACGT. This restriction enzyme recognizes the DNA sequence CCCGGG. It cuts the DNA strands between the bases C and G. The resulting DNA fragments have no single-stranded sticky ends.
Once a gene has been isolated using a restriction enzyme, we often need to make copies of that gene. Copies are created using a process called DNA cloning or gene cloning. Gene cloning is used to make multiple copies of a gene. We call this amplifying a gene or amplifying the DNA. The foreign DNA is going to be inserted into a plasmid, and that recombinant plasmid is then going to be inserted into a bacterial cell. Here we see a diagram showing how DNA cloning takes place. So if you're looking at this picture right here, we take our gene of interest out of a cell. That gene of interest is removed using a restriction enzyme. A plasmid is then cut open using the restriction enzyme, and that gene of interest is inserted using the enzyme DNA ligase. So here you see a recombinant plasmid, a plasmid that contains a gene from another source. That plasmid could then be inserted into a bacteria using the process of transformation. We know in order to perform transformation, we have to heat shock the bacteria. By heat shocking the bacteria, it's going to open specialized surface proteins that allow that bacteria to take in the plasmid. Once that bacteria has taken in the plasmid, the bacteria is going to reproduce. Every time the bacteria reproduces, it is also going to replicate the plasmid. As the bacteria is reproducing, it's also constantly expressing the genes on the plasmid and creating the proteins. So from one bacteria, we can make thousands of bacteria that each contain the gene of interest and contain many copies of the protein that is trying to be made. From those bacteria, we can do two things. We can isolate the genes that have been copied, or we could harvest the proteins that have been made. Here you see some examples of proteins that have been harvested using DNA cloning. One protein that is made in mass quantities through DNA cloning is insulin. Insulin is a protein given to diabetics. Another protein that is harvested using DNA cloning is human growth hormone. Growth hormone is given to children that have stunted growth. And another protein created through DNA cloning is a protein called TPA. TPA is given to people that have had heart attacks, and it's going to dissolve blood clots. Here you see some examples of genes that we can harvest from the bacteria. We can mass produce genes that are then given to plants that allow them to be resistant to pests. We could also use those genes and insert those genes in other bacteria. Today, an important thing that we're doing to bacteria is that we're genetically engineering them to be able to clean up toxic waste. In this case here, we see the cleanup of an oil spill. These bacteria have been genetically engineered to consume the oil to break it down into a harmless waste product. This process of using bacteria to clean up toxic waste is called bioremediation. So you can see here through the process of DNA cloning, bacteria are used to create massive quantities of genes and massive quantities of the proteins that they code for. These genes and these proteins can be harvested and they can be used for practical purposes. Another way to copy DNA other than using gene cloning is a process called PCR. PCR is typically used when the DNA source is scanty or impure. PCR can be used when DNA has been found at a crime scene. And PCR is also going to be used to amplify DNA. It's going to be used to make many copies of the DNA. PCR basically mimics the process of DNA replication. Let's see how it's done. So if you're looking at this picture here you see our targeted sequence of DNA. Here's our gene that we want to make copies of. In the first cycle of PCR, 3 things need to take place. Number 1 is denaturation. We have to open up the 2 strands of DNA. This is done by heating up the DNA. By heating up the DNA, we break those hydrogen bonds holding the 2 strands together. The next step is called annealing. In the process of annealing, primers are going to be added. Once those primers are added, then we have our third step, which is extension. The enzyme DNA polymerase is added. Polymerase is going to come in and it's going to extend the primers, making the new strands of DNA. You can see that each cycle of PCR has 3 processes: denaturation, annealing, and extension. Denaturation, heating up the DNA to open the 2 strands. Annealing, adding primers. And extension, DNA polymerase synthesizing the new strands of DNA. At the end of our first cycle of PCR, we go from having 1 gene to having 2. The second cycle of PCR is going to create 4 molecules of DNA. The third cycle of PCR is going to create 8 molecules of DNA. You can see that as PCR progresses, these extra little pieces that we are not looking to copy are slowly removed. You can see that by the third cycle, we have 2 molecules of DNA that no longer contain these extraneous pieces. They only have the selected genes. PCR stands for polymerase chain reaction, and it's a way that we synthetically go through DNA replication in the lab. And it's used to amplify DNA to make many copies of a gene or a piece of DNA. Once a gene has been isolated, we can analyze that gene using a process called gel electrophoresis. We're going to perform this process in a lab that we performed this year. When restriction enzymes cut a piece of DNA, it's going to create fragments. These fragments are called restriction fragments. They're fragments of DNA created by cutting DNA with a restriction enzyme. These fragments will then be separated by size using this process of gel electrophoresis. In the process of gel electrophoresis, these fragments of DNA are loaded into wells contained within a gel. Here you could see our gel, and the name of the gel that we use is called Aragos gel. And Aragos gel is going to have pores in it. It's going to have tiny holes in it that it's going to allow the DNA to travel through the gel. Once the DNA containing the restriction fragments has been loaded into the wells, the gel is hooked up to a power source. And you can see from the power source, you're going to have one side of the DNA have a negative charge. One side of the gel have a negative charge, and the other side of the gel have a positive charge. So this gel is going to contain an electric field. It has an electrical current running through the gel. Since DNA is negatively charged, it's negatively charged because of the phosphate groups, the DNA is going to travel from the negative side, where the wells are, over to the positive side. The smaller fragments are going to be able to travel through the pores much easier than the larger fragments. So when we kill the power source and turn it off, the shorter fragments of DNA will have traveled very far, and the longer or larger fragments of DNA have traveled a much shorter distance. You can see here that this process of gel electrophoresis is going to separate out all of our DNA fragments based upon their size. Once this has been done, we could then analyze those specific fragments of DNA. Every individual, when their DNA is cut using a restriction enzyme, is going to create fragments of slightly different lengths. This is because of polymorphisms. Polymorphisms are slight differences in DNA sequences from individual to individual. These fragments that are created by cutting an individual's DNA with a restriction enzyme are called RFLPs, restriction fragment length polymorphisms. These RFLPs are going to be used to create a DNA fingerprint. They are unique to every individual and can be used to identify that individual. Usually the part of the DNA that we choose to cut using the restriction enzyme is a part of the DNA called a short tandem repeat, STRs. These short tandem repeats are non-coding regions of DNA that have repetitive DNA, and the number of repeats is highly variable from person to person. No two people will ever have the same number of repeats, have the same sequence of DNA. Therefore, when you cut this region of DNA using a restriction enzyme, every person's RFLPs are going to have a different length. These different sized RFLPs created by cutting the short tandem repeats is going to be unique from individual to individual. You can see here that we can compare these RFLPs to do a couple different things. One thing that we can do with these RFLPs is create a DNA fingerprint. It can be used to identify an individual. This is done in crime scene investigations. This is also done in cases of paternity testing. Here we look at some gels that were created using DNA from different individuals. In this first gel, we have the mother's DNA run, we have the child's DNA run, we have suspected father 1, and we have suspected father 2. A child is going to share all of their fragment lengths, all of their RFLPs. They need to be shared in common with both their mother and their father. So if you look at the child got this RFLP here from his mother. That means that these 3 other RFLPs need to come from the father. Now if you look at this first one right here, both suspected fathers have this band. But if you look at suspected father number 1 right here, these 2 bands don't line up. You can see that father 2 contains the other 2 bands. That means that suspected father number 2 definitively is the father of the child. If you look at the second example right here, here you have Mary, Bob, Larry, and the child. Here you can see the band shared in common with mom. And now that you understand how to do it, you can see that this child obviously belongs to Larry, because Larry is going to share the other bands in common with the child. You can see that every individual has a unique banding pattern, and that you can see a child is going to share half of its bands with its mother, half of its bands with its father. This is done in cases of paternity testing, This can also be done to identify an individual in a crime scene. Another thing that we can use gel electrophoresis to do is we can use this to determine if an individual is homozygous for a gene or is heterozygous for a gene. If we run two alleles through gel electrophoresis, if they have the same banding pattern, we know that that individual is homozygous. However, if they have a different banding pattern, we know that that individual has to be heterozygous. Remember, if an individual is heterozygous, they're going to have two different alleles for a gene. If they have two different alleles, they have two different sequences of DNA. Two different sequences of DNA are going to be cut using a restriction enzyme in different locations. By cutting the DNA in different locations, it's going to create different restriction fragment lengths. Therefore, when you run the two alleles through gel electrophoresis, each is going to make a unique banding pattern. This is how we test the DNA to identify if a person is heterozygote or if a person is a carrier for a specific genetic disease. Often when a sequence of DNA is run through gel electrophoresis, we're going to end up having many different fragments. If you look at a sequence of DNA cut with the restriction enzyme, creating many fragments, you can see that gel electrophoresis is going to separate out all of those fragments. Now, what if we want to isolate specific genes of DNA? We don't want to look at every fragment. We only want to compare very specific fragments. In order to isolate these genes or these fragments, we're going to use a process called Southern blotting. Southern blotting is going to combine gel electrophoresis with a process called DNA hybridization. In DNA hybridization, we are going to use a radioactive probe in order to isolate a selected gene. So if you look here you see our selected gene that we want to isolate. The radioactive probe that we create is a piece of single-stranded DNA that is complementary to the selected gene. Since it's complementary to the selected gene, it's going to base pair with that gene. You'll see in a second that we could then use this radioactive probe in order to isolate that specific gene. Here you see a diagram showing the process of Southern blotting. It begins with gel electrophoresis. The DNA and the restriction enzymes are combined together, creating restriction fragments, pieces of DNA of all different sizes. Those fragments are then sent through gel electrophoresis, and it's going to create many different bands depending upon the size of the fragments. These fragments furthest away are going to be smallest. These fragments that are close are going to be much larger. Once the gel is completed, we are going to put a piece of nitrocellulose paper on top of the gel, and then on top of the nitrocellulose paper, we're going to put a stack of paper towels. That stack of paper towels is going to draw all the water and all of the fragments of DNA out of the gel through the process of capillary action. So through capillary action, the DNA is going to be transferred from the gel to the piece of nitrocellulose paper. That piece of nitrocellulose paper is then given the radioactive DNA probes. These are going to be the fragments of DNA that are complementary to the fragments that we are looking to isolate. Each of these probes is going to base pair with the specific fragments that we want to isolate. Once we allow the probe to react with the fragments of DNA, a piece of film is placed on top of the nitrocellulose paper. Those radioactive probes are going to burn an image into the film. This film is only going to contain bands from those specific genes or those specific fragments that we wanted to isolate. So if you look here in the gel, you see many different fragment lengths. In our film, the end result, we only have those specific fragments or those specific genes that we wanted to isolate. This was accomplished by adding a radioactive DNA probe. This radioactive DNA probe base paired to all the specific genes that we wanted to isolate, and the radioactive probe burned an image onto the film. We can also use DNA technology in order to determine the structure of a gene, in particular to determine the sequence of bases in a gene. This process, called DNA sequencing, is accomplished by using modified nucleotides called DDNTPs. These stand for dideoxyribonucleotides. This process, which is going to utilize the dideoxyribonucleotides to sequence a gene, is called dideoxy chain termination method. These DDNTPs are very special nucleotides. Here you see 4 of them in the upper right hand corner. And 2 things happen with these DDNTPs. Number 1, each one is fluorescently tagged with a different color. You can see here the adenine is purple, the cytosine is pink, the thymine is yellow, the guanine is green. When these DDNTPs are used during the process of DNA replication, they are going to end the growing strand. Whenever they are inserted by the enzyme DNA polymerase, replication stops. It stops because they lack the attachment site for the next nucleotide. In order to accomplish DNA sequencing, we are going to undergo the process of DNA replication. So if you look at the top of this diagram here you see our gene that we want to sequence. I know here it gives us the sequence of bases, but our goal is to determine what that sequence is. Here you see all the different components needed to replicate the DNA. We have our primers, we have DNA polymerase, we have all our normal nucleotides, and now we have our dideoxyribonucleotides. All of our nucleotides that are fluorescently tagged and that will end the growing strand of DNA. The template strand of DNA, or the gene that we want to sequence, is artificially put through the process of replication. And you can see here that DNA polymerase is going to pick from the nucleotides to create the growing strand. Whenever polymerase picks one of the dideoxyribonucleotides, though, that new strand of DNA is going to end. And that last base, that dideoxyribonucleotide, is going to be fluorescently tagged. Now I know this picture here shows all these new fragments created going in order from smallest to largest, but in reality, as the DNA is being replicated, DNA polymerase is randomly going to choose from one of these deoxyribonucleotides at a random point, and it's going to create all these different fragments that have all these different sizes. However, each fragment is going to end with one of these fluorescently tagged deoxyribonucleotides. Once a whole mess of these fragments have been created, these fragments are going to be run through gel electrophoresis. We know that gel electrophoresis is going to separate out each individual fragment based upon its size. The smallest fragments are going to travel the furthest, and the largest fragments are going to travel the shortest distance. When gel electrophoresis ends, you're going to have a series of fragments, and each fragment is going to have a color associated with it. You can see that the smallest fragment right here is going to end with C, the cytosine, and here you could see the pink fragment right here. The next one is going to end with guanine, which is green, and here you see guanine right there. So by looking at the sequence of the colors in the gel, we then determine the sequence of the nucleotides making up the gene. This was the method by which we've determined the sequence of all the bases in all of the genes in our genome. All right, what can we use DNA technology to do? One of the things that we're working on DNA technology to do is to cure disease. A method of curing disease using DNA technology is called human gene therapy. The process itself has merit, however, it has not been largely successful so far. It's only been successful in treating a couple of genetic diseases. Let's look at how gene therapy works. Gene therapy can only be effective at a disease that's caused by a single defective gene. These would be diseases that are caused by being homozygous recessive. We've talked about a bunch of these, like cystic fibrosis, stuff like that, Tay-Sachs. Now, we know that if the person is homozygous recessive, both recessive alleles code for non-functional proteins. So scientists said, let's just insert the dominant allele. If you insert the dominant allele, you make the normal functioning protein. In order to insert this dominant allele, we are going to use a virus as the vector, as the way to insert the gene. In particular, we're going to use a retrovirus. We know that retroviruses have RNA as their genetic material and not DNA. So we begin by inserting the RNA version of the normal allele into the retrovirus. So here you see the retrovirus, here you see its RNA, here you see the selected dominant allele that we want to insert into the individual. We then extract cells from that individual. We don't just extract any cell, we need to extract adult stem cells. Adult cells that are going to continue to divide throughout that individual's lifetime. Usually these adult stem cells are extracted from the individual's bone marrow inside of the bones. So some of these cells are extracted, and then the retrovirus is then given to the cell. The retrovirus infects the host cell, which is a bone marrow cell from the patient. We know that when retroviruses infect host cells, inside the host cell, the viral RNA is converted into DNA. That viral DNA then becomes a provirus. A provirus inserts itself into the host cell's genome. So by doing this, we've effectively taken that dominant allele and incorporated that dominant allele into the individual's DNA. These cells that now contain the dominant allele are injected back into the patient's bone marrow. These cells that have been genetically engineered to have the dominant allele will now continue to divide throughout that person's life. They will continue to make that functional protein and hopefully cure them of the disease. Like I said, this process has merit to it. However, it has not been largely successful. It's only been successful in a couple of different genetic diseases. Here you see another example of how gene therapy works, right? That gene is inserted into the virus, the retrovirus. You can see that that retrovirus then infects the host cells that have been extracted from the individual's bone marrow. These are going to be adult stem cells. And then these genetically engineered cells are reintroduced back into the individual. How else can we insert genes into eukaryotic cells? We know that it's very easy to insert genes into prokaryotic cells because prokaryotic cells take in plasmids easily through the process of transformation. How do you then insert genes into eukaryotic cells? We often use a process called extraporation, which you see right here. In this process of electroporation, or however you pronounce it, you apply a brief electrical pulse to the cell, and that's going to create temporary holes in the plasma membrane. Once these temporary holes have been created, that cell then takes in the foreign gene. Another method to insert genes into eukaryotic cells is to inject the DNA using microscopically thin needles. We'll see in a minute that this is done to insert genes into egg cells. Once inside the cell, once it goes in through the process of electroporation or using a thin needle, that DNA is going to incorporate itself into the host cell's DNA through natural genetic recombination. So what can we do with these inserted genes? Here we look at how genetic engineering is used in agriculture. We can look at animal husbandry and quote-unquote farm animals. Animal husbandry is the process of selectively breeding animals. Now, we don't think of selective breeding as a form of genetic engineering, but it is. When you choose what animals mate, in essence what you're doing is you're choosing what genes are being passed down from generation to generation. We are also today creating farm animals. These farm animals are transgenic. They are animals that contain genes from other sources. These are animals that have been genetic engineered. You can see that we can genetically engineer animals by inserting a cloned gene into an egg cell. This is done using that microscopic needle that we just talked about. Since the gene is inserted into the egg cell, as that animal grows and develops, that gene will be contained in every single cell of that animal. This is being used today to create animals that are leaner, that are more muscular, or that have better wool. Right here you see a bull that has been genetically engineered to be extremely muscular. But we can also do this to make animals pharmaceutical factories. We could do this in order to have the animal mass produce this protein that we're going to be looking to extract later on. Finally, today we are also genetically engineering plants. We genetically engineer plants in order to make them resistant to things like insects, to microbes, and other pests. And this improves our crop yield. It decreases the number of plants that have been consumed by other animals, or that have infections, or that have been eaten by insects. The process used to insert genes into a plant is pretty interesting. We're going to actually use a plasmid called the TI plasmid. This is a plasmid that is taken from a particular type of bacteria called Agrobacterium. This Agrobacterium has the TI plasmid. That TI plasmid is opened up using a restriction enzyme. That gene of interest is inserted into the TI plasmid using the enzyme ligase. That TI plasmid is then directly introduced into the plant cell very early in its development. Now we know that plant cells do not normally contain plasmids, so we are artificially inserting this plasmid into the plant cell. The TI plasmid is unique because once it has been inserted into the plant cell, it is going to take that gene and it's going to insert that gene of interest into the host cell's genome. So now when that plant reproduces, when those cells undergo cell division and they create an adult plant, every cell inside that plant is going to contain that gene, allowing it to become resistant to insects or to bacteria or whatever trait we want that plant to express.